Just so you know, we will take discussion at the end of the session. Our next speaker is Dr. Robert Margolskee, who's going to be discussing taste receptors in the gut and how they influence eating behavior. Dr. Margolskee is the Associate Director of the Monell Chemical Senses Center, where he joined the faculty in 2009. Dr. Margolskee's long-standing research focus is on the molecular mechanisms of taste transduction, utilizing molecular biology, biochemistry, structural biology, electrophysiology, and transgenesis to study the mechanisms of signal transduction in mammalian taste cells. More recently, he's been studying the chemosensory functions of taste signaling proteins in the gut and pancreatic endocrine cells. Dr. Margolskee has made numerous seminal discoveries in the taste field, including identification and the molecular cloning of taste-specific receptors and G proteins. Dr. Margolskee.
Thank you, Danielle, and I'd like to thank the organizers of this very interesting meeting for the invitation to speak to you today. And what I will be covering are taste receptors in the gut, and before getting to that, a little bit of an overview of taste receptors in the oral cavity, and then describe newly identified roles of these same receptors in the gut and their newly developing functions in digestive physiology. And then I'll circle back to the oral cavity to describe some of the stunning parallels between the function of taste receptors in the oral cavity and in the gut. So just a quick overview of the oral taste receptors found within taste cells in the tongue. This is a diagram of the human anatomy of the taste system, and you'll find taste buds scattered throughout the oral cavity, the front, sides, and back of the tongue, and then in the lower right-hand portion of the slide is a cross-section of the circumvallate papillae at the back of the human tongue. There are about a dozen of these papillae structures, and in the side walls of the papillae you will find the taste buds, which are collections of 50 to 100 specialized epithelial cells. They're not neurons, but they have a number of neuronal properties, and at the apical end of the taste bud, you'll find these finger-like microvilli processes, and contained within these microvilli is the transduction machinery for the taste cells, where you'll find the receptors, G proteins, and associated ion channels and second messenger enzymes involved in taste transduction. So we've come to realize that there are a number of different types of taste receptors in the oral cavity, and underlying the taste response in each of these different types of cells is a different molecule or multiple molecules that act as transduction channels and transduction receptors. And over the last decade or so, work from a number of laboratories, including my own, Linda Buck, and Charles Zucker and Nick Reba, has together identified a number of these taste quality receptors. So we know that for bitter, for example, we have a family of 25 to 30 G protein coupled receptors called the T2Rs, the type 2 taste receptors. These are fairly discrete 7 transmembrane receptors that couple to G proteins, and they don't have much in the way of extracellular or intracellular elaboration. That contrasts with the sweet receptors, which is a dimeric or multimeric combination of T1R2 and T1R3, type 1 receptor 2 and type 1 receptor 3, which together will respond to a number of sweet compound sugars and non-caloric sweeteners. And a related receptor, which is a combination of T1R1 and T1R3, will respond to umami, amino acids, including monosodium glutamate, and a taste that can be described as savory of peptides and proteins. Now, for sour and salty, the transduction channels are less well understood. ENaC certainly plays a role in a salty taste, but that is more for low concentrations of salt. There's likely to be at least one other transduction channel out there for high concentrations of salt. And sour has a number of candidate channels, including acid sensing ion channels and HCN in the PKD family members, but no definitively identified transduction channel as yet for sour tastes. Now, it has come to be understood in the past few years that not only are there these taste cells and taste receptor molecules in the oral cavity, but there are also taste-like cells in the gut. And these are not actual taste cells, but they have a number of characteristics that are very much in common with the true oral taste cells. And that is that they have the morphologic appearance under light and electron microscope of oral taste cells. They also express many of the same taste signaling proteins that are found in the oral taste cells, the taste receptors, the G proteins, and the ion channels. And they are found in a specific subset of gut cells, particularly gut endocrine cells, as well as chemosensitive brush cells. And what we're coming to understand is that they do play a chemosensory role in responding to nutrients during the digestion process, and they do play important developing role in integrating physiologic responses during different stages of digestion. So here's a schematic diagram on the left of a taste cell from the oral cavity and the transduction pathway as we've come to understand it over the past 10 plus years. In this oral taste cell, you have G protein coupled receptors at the apical surface. They couple to G proteins, including the taste G protein gusteucin. This initiates a signal transduction cascade, which can activate multiple signaling enzyme, release various second messengers, including inositol trisphosphate, and that can lead to activation of particular channels, including the calcium-activated TRP channel, TRPM5, leading to ion influx, depolarization of the taste receptor cell, and then neurotransmitter release, which can either reach the gustatory afferents, or in this case, it actually is a lateral form of transmission from one type of taste receptor cell in the oral taste bud to another. And it turns out we're having very similar kind of process going on in certain subsets of the endocrine cells in the gut, particularly the L and K cells that express GLP-1 and GIP, and the LK cells that express both of these peptides. And we're finding, and I'll show you this data in just a moment, a number of the same taste receptors, the taste G protein, the same second messenger enzymes and second messengers, and this TRPM5 channel are found to be present both in the oral taste cells and these taste-like endocrine cells in the gut. And one of the differences is that instead of a bona fide neurotransmitter, more often we appear to be leading to release of a neuropeptide hormone, such as GLP-1, which would then make its way either to the bloodstream or locally for paracrine or true hormonal effects, or reach the local nerve fibers to have a neuronal effect. So the idea that there are taste signaling molecules in the gut dates back to Dirk Hofer's work in Drenkhan's laboratory in 1996, when he identified that the G protein alpha-gusteucin is expressed in cells that have the appearance of taste receptor cells in the stomach and the small intestine. Subsequent to Hofer's work, Rosengert's group has identified a number of the T2R bitter taste receptors as being present throughout the gut, in the stomach and small intestine, as well as in a number of gut cell lines, including gastric enterochromaffin cells and IEC6 cells, and subsequently Soraya Shirazi-Beachy by PCR has identified T1R receptors as being present in the gut. Now, in collaboration between my group and Soraya Shirazi-Beachy's group, we carried out more detailed microscopic studies to identify actually which cells within the gut would express taste receptor proteins. And the sections shown here are tissues from human duodenum, and what we see by in situ hybridization in the left panel is that T1R2 and T1R3, the two components of the sweet receptor, are present in a small subset of cells lining the small intestinal mucosa. And these cells have the typical appearance of enteroendocrine cells, and we've confirmed this by immunohistochemistry with antibodies directed against both subunits of the sweet receptor. And then in somewhat more detailed studies that were carried out in collaboration between my group and that of Josephine Egan at the NIH, we identified in both human and mouse tissue that several taste signaling proteins, in fact, essentially the entire taste transduction pathway as we knew it in taste cells, was present also in gut endocrine cells, and particularly to be found in the L cells that also express GLP-1. So here you can see in green a number of taste signaling proteins, alpha-gusteucin, its beta and gamma partners. They're co-expressed with GLP-1 in the L-type enteroendocrine cells. We also have expressed PLC beta-2, TRPM5, and T1R3. So it does appear that the entire taste signaling transduction cascade can be found in certain cells within the gut in this tissue specimen from a human, but similar results from the mouse. And more recently, Yan Li in my laboratory has undertaken a study to examine co-expression of gusteucin positive endocrine cells from the duodenum, jejunum, ileum, and then the colon, and co-expression of this taste-like cell marker gusteucin with GLP-1, GIP, and both of these hormones, and found that in colon there is a roughly equal level of LK and LK co-expression with gusteucin, but in the duodenum the L cells are actually rather small in number, and it's mostly K cells in the GLP-1, GIP co-expressing LK cells that are expressing gusteucin. Yan went on to look at co-expression with gusteucin in the gut and in the colon, found that a number of short-chain fatty acid receptors were co-expressed with alpha-gusteucin in endocrine cells in the colon, and this would include GPR43 and GPR41 being co-expressed with alpha-gusteucin, and to look at what potential physiologic role there might be for gusteucin in the colon. She turned to gusteucin knockout mice and determined that in a tissue preparation ex vivo, wild-type colon tissue would be stimulated by a couple of short-chain fatty acids, acetate and butyrate in particular, to release GLP-1 into the cultured medium. But in gusteucin knockout mouse, we would lose a dose response effect of the short-chain fatty acids, implicating gusteucin in these colon endocrine cells as connecting the detection of short-chain fatty acids to the efferent output, the effector GLP-1. One other short vignette I'd like to relay to you from the gut is work that was done a few years ago in collaboration between my lab and Soraya Shirazi-Beachy. Here we show a cross-section of the small intestine, and I draw your attention to SGLT1, sodium glucose cotransporter 1. SGLT1 is typically the rate-limiting step for glucose uptake. This is a co-transporter protein that co-transports glucose and sodium from the gut lumen across the absorptive enterocytes, and then in the basal aspect, passive diffusion by various glucose transporters will lead to release of the sugar into the bloodstream. What we looked at with Soraya Shirazi-Beachy's group was the effect on SGLT1 level expression of a couple of knockout mice. So in the top panel are experiments done with wild-type mice, looking at messenger RNA, protein in the middle panel, and then glucose uptake activity. These are animals that were treated with either a low-carbohydrate diet or a high-carbohydrate diet. And in wild-type mice, we see that there is basal level of SGLT1, which will increase significantly on a high-carbohydrate diet. And this mRNA increase is associated with a protein increase for SGLT1 and also with an increase in glucose uptake activity. The situation is rather different when we look at two different knockout mice, either mice that are missing T1R3, the component of the sweet and umami receptor. And what you see is on either the low or the high carbohydrate diet, equal amounts of messenger RNA, protein, and glucose uptake. So there is no change in SGLT1 levels in response to the dietary manipulation of adding high carbohydrate. And it's very much the same effect. I'm sorry, I may have misspoke. This set of panels here is with the gusteucin knockout. The lower set of panels is with the T1R3 knockout, and they really are indistinguishable. So you require two taste signaling components, the G protein gusteucin and the taste receptor component T1R3 to elicit this increase in SGLT1 and thereby increase the amount of glucose uptake activity. And then we find actually a similar effect If we look at a low-carbohydrate diet, which has been supplemented with a non-caloric sweetener, in this case sucralose, the mice were given sucralose in their water bottles, and you see in wild-type mice, low carbohydrate versus low carbohydrate plus sucralose, you get, in the presence of the sweetener, an increase in SGLT1 messenger RNA, an increase in the protein, and not shown here, but also an increase in the glucose uptake activity. And then in either one of the knockouts, the gustosine knockout or the T1R3 knockout animal, the addition of sucralose has no effect on SGLT1 messenger RNA or protein or glucose uptake activity. So this would indicate that there is a chemosensory detection pathway in the gut responding to luminal sugars and luminal sweeteners that would lead to a signal to upregulate SGLT1 and increase glucose uptake activity into and across the gut. So going a little further into the gut, we've recently found, as have others, that there are these taste-like cells in the pancreas. From our work, we've identified the taste G protein gustosine as being expressed in alpha cells, shown here by staining for glucagon, and not to be present in beta cells, which are stained positive by insulin expression. However, if we look at expression of T1R3, we find that the sweet receptor, umami receptor component is expressed both in the glucagon-positive alpha cells as well as in the insulin-positive beta cells. And as far as what function these taste receptor subunits and gustosine might have in the pancreas, this is an ongoing series of studies. The slide here and the next slide I'll be presenting are from in vitro studies with islet transplanted tissues. And what we find is that the basal insulin secretion from islets from wild-type mice can be stimulated by the addition of a non-caloric sweetener, sucralose. So there is an enhancement of insulin release from the islets, and we do have some evidence that this same sort of result is going on in vivo in the mouse. And if we look at the response in wild type, in the white bars, versus a T1R3 taste receptor knockout, we see that another sweetener, a non-caloric sweetener, SC45647, in wild type will give a dose-dependent increase in insulin release. But in the knockout animal, you completely lose this response from the sweetener-enhanced insulin release. And we can mimic reasonably closely the same effect by adding germarin, which is a inhibitor of the mouse taste receptor, the sweet taste receptor. And adding germarin will mostly block the sweetener enhancement of insulin release. So to just wrap up my conclusions for this portion of my presentation, we've identified whole taste signaling pathways in the gut, throughout the gut, in fact, from proximal to distal gut, also within the pancreas. We've identified these taste elements in the L and K and LK cells. Gustosine, T1R3, both are involved in GLP-1 and GIP incretin release in response to sweeteners. And in the proximal gut, there is involvement in mouse models of both gustosine and T1R3 in regulating SGLT1 glucose transporter activity. In the colon, In contrast, there is implication that gusteucin is involved in release of GLP-1 and GIP in response to short-chain fatty acids, which could be released during the digestion process and reach these endocrine cells in the colon. As far as in the pancreas, the role of T1R3 and gusteucin, this is an evolving story, but preliminary evidence would argue that there is the ability of these expressed taste receptors to detect sweeteners and, under some circumstances, to synergize with and promote insulin secretion from the islets. So I'd like to circle back in the remaining few minutes to the oral taste cells and describe some of the gut-like properties of the oral taste cells and how perhaps we should consider the digestive tract from the oral cavity to the distal colon as one integrated system with specialized variations and specialized properties depending upon which part of the alimentary or digestive system we're talking about. But for now, let me come back and describe some of our findings most recently about oral taste cells. We found that a number of gut hormones, GLP-1, GIP, CCK, are expressed in the taste cells. They have been found in multiple subtypes of the taste cells, and we're finding in some cases at least release of hormones directly from the oral taste cells into the bloodstream in response to sugars and sweeteners. And then most recently, we've identified intestinal type sugar centers, SGLT1, glucose transporters, and the like to be present not just in the gut but also in the oral taste cells, and pancreatic type metabolic sensors such as the ATP-gated potassium channel as likewise being expressed in the oral taste cells. So backing up just one moment, this is another schematic diagram of the sweet taste receptor. Remember, it's a heterodimeric or multimeric combination of T1R2 and T1R3, these two family C G protein coupled receptors, and how this single receptor can respond to multiple sweet compounds is because it has multiple ligand binding domains. The extracellular amino terminal domain of T1R2 and T1R3 can respond to sugars and small molecule sweeteners such as aspartame. There is a cysteine-rich linker region that ties together the amino terminal domain with the 7 transmembrane helices and peptide or protein sweeteners such as Brazein will interact with this portion of the sweet taste receptor. And then in the transmembrane domain of T1R3, small molecule sweeteners such as cyclamate and a sweet antagonist lactosol will interact with the transmembrane domain of T1R3, and a different sweetener, perlitine, can interact with the transmembrane domain of T1R2. Now, it had been proposed that T1R2 and T1R3 was the only sweet taste receptor, and this was based on in vitro expression studies and mouse knockout studies from the Zucker and Reba group. We, in my group, with a different knockout that we had generated, a T1R3 knockout animal, found something somewhat different. We found that in comparison to wild type, indeed, there is a major loss in these nerve responses. And here, this is just a diagram of the chordae tympani nerve responses from wild type in the bluish color, versus T1R3 knockout. And for non-caloric sweeteners such as saccharin, we see a major difference. SC45647, sucralose, also a major difference. We've lost almost entirely the chordotympany nerve response to these non-caloric sweeteners, and actually we've lost most of the response to sucrose and to D-tryptophan, which are two caloric sweeteners. But where we differ in our results from the Zucker and Reba group is that the sugars, glucose, maltose, and somewhat fructose, and the sugar alcohol, sorbitol, do not have major differences between the wild type and the knockout, arguing that T1R3, and because it's an obligate heterodimer, T1R3 in combination with T1R2 is indeed likely to be the primary or only receptor for the non-caloric sweeteners like saccharin and sucralose. But there likely is something else in the oral cavity that's functioning to respond to the sugars. So we set out to identify what this mystery transduction pathway might be. And our initial hypothesis was that there would be a gut-like detector functioning in the taste cells, much as we were following taste-like function in the gut. So here's a diagram of the functioning in pancreatic beta cells, where glucose in the bloodstream will get taken up into the pancreatic beta cells, undergo glycolysis, increased level of ATP will bind to the SIR1 component of the ATP-gated potassium channel, closing this channel, depolarizing the pancreatic beta cells, leading to calcium influx and insulin exocytosis. So our hypothesis was a very similar pathway would be functioning in taste cells, and using various techniques, we found a number of these components were indeed present in taste cells. Glucose transporter subunits, GLUT8, GLUT9, SGLT1, and components of the KATP channel, SIR1, KAR 6.1, were all present by RT-PCR in taste tissue versus non-taste tissue control. By immunohistochemistry, we confirmed expression of a number of the glucose transporters, GLUT4, SGLT1, SIR1 are all in taste tissue. And perhaps most revealingly, when we mark the taste cells by expression of a transgene, green fluorescent protein, in the T1R3 sweet umami responding cells, we see a great extent of co-expression of these glucose transporters in KATP in exactly the same subset of taste cells in the oral cavity. Let me skip this for the sake of time and summarize this portion of the talk, that several glucose transporters as well as the KATP subunits are present together in the sweet responding subset of taste cells. So here you have T1R2, T1R3, the canonical sweet receptor for taste cells, but co-expressed in the same cells are glucose transporters, KATP subunits, and SGLT1. And we think that these can work together additively, perhaps synergistically, or alternatively to provide either independent or interdependent sweet transduction pathways, and might provide a mechanism whereby people and animals could distinguish caloric from non-caloric sweeteners. So I think time does not permit to go over these next few slides. Perhaps in the discussion we'll get back to it. And I'd just like to acknowledge support from the NIH, both NIDDK and NIDCD, and a number of my colleagues who have carried out these studies. Thank you.